Hello. I'm audible to everyone. Hello. Hello. Am I audible to everyone? Hello.
Yes, sir, you are audible.
Oh, thank you. So we'll start with this. A warm welcome to all the respected dignitaries, esteemed speakers, sir, dear participants to day three of our faculty development program. I hope you all are having an insightful and enriching journey so far. I welcome today's speaker, sir, Dr. Devarshikar Mahapatra, sir. Sir has hold master degree in pharmacy from Guru Ghashidas Vishwavidyalaya, Central University, Bilaspur, and sir has earned PhD from H. Hari Singh Gaur Vishwavidyalaya, Central University, from Sagar, Madhya Pradesh. Sir served as an ICMR Senior Research Fellow and gained significant research experience at the Council of Scientific and Industrial Research, CSI, Hyderabad. Dr. Debarshri, sir, has a teaching experience in medicinal chemistry, computational chemistry over past 10 years of academic research, 5 years of active monitoring, and 2 years in administrative role. Dr. Mahapatra, sir, research interest has computer-aided drug design, synthesis of low molecular weight ligands, targeting biologically relevant proteins, formulation development, and explorations of natural bioactive compounds. Sir has authored more than 100 research articles, indexes in Scopus, and presented his original findings in various international publications conferences. Sir has been listed among the top 2% scientists worldwide for 5 consecutive years from 2020 to 2024, as per the global database published by Stanford University, USA. Sir has been honored with the Young Investor Award by the Bill and Melinda Gates Foundation, USA. I welcome all of you once again, and I request Speaker Sir to start the sessions of Day 3 Faculty Development Program. Thank you all.
Oh,
Devashi Sir, you can share your screen, sir.
then you are not audible. No.
Sir, having any issue? Yes, sir, you are audible, sir. Now.
ചെയ്താൽ മതി പറഞ്ഞിട്ടാണ് അതിന്റെ രീതിയുണ്ട് അല്ലോ അതാ ഒരു മിനിറ്റ് അപ്പൊ നമ്മളുടെ 43 വരത്തില്ലേ അതിലെല്ലാം ഉണ്ടെങ്കിൽ സ്പ്ലിറ്റ് ചെയ്യായിരുന്നു ഇൻഡിവിജ്വലി ക്ലാസ്സിലൊക്കെ
Thank you all. Please mute yourself.
രണ്ട് കണ്ട ഇതാ ഞാൻ പറയുന്നത് 8 ഇങ്ങനെ വരരുതല്ലോ
അത് പലതായിട്ടാണ് വന്നിരിക്കുന്നത്. അതായത് ചില PDF തുറക്കുമ്പോൾ 8 എണ്ണം, ചിലതിൽ 2 എണ്ണം.
Is it audible? Is it audible now?
Yes, sir.
Is it audible?
Yes, sir, it's audible, sir.
Yes, sir.
Okay. Okay, then it's fine. Then it's fine. Okay, thank you. So I think you can see the slides. Please response.
Yes, sir.
Okay, so let's
Yes,
start.
sir.
So in this today's session,
We can
that
see
is
the
the
screen also.
tools and techniques for this systematic literature review and bibliography. So in this 4th to 8th of August of this FDP, that is organized by the Haldia Institute of Pharmacy. So here in the 3rd day, so this will be one of the most important topic where I will be providing you an overview of all the tools and the techniques that we are going to see that have been applied in applications for the systematic literature review, as well as in the bibliographic part, we will see whatever the softwares and whatever the plugins that we generally use for the bibliographic representation and the citation. So myself, Dr. Devarshikar Mahapatra from Chitkara University, Rajpura, Punjab. So today I will be providing you an overview. This session I want to be not just as a lecture, but as an interaction from both the sides. So now, since this is a FDP and there are a lot of other participants, I hope, who have been into research recently as a research scholar, as well as there are faculties who are already been pursuing their PhDs. Okay, so therefore, this topic remains one of the most important portion while in the selection of the topics, first of all, and secondly, that how to effectively conduct a literature. Okay, how we can effectively conduct a review of a literature, because literature is very, very diverse. Okay, so there are a lot of databases. There are a lot of databases, and uh, I can see when in a current knowledge, you can see a lot of gaps. Okay, and that gaps will be the key areas, or I can say the hot areas, where a researcher or a scholar can do their research and continue with the new findings. So here I can tell you that what is the basic importance of the literature review. So it will help in the identification of your gaps. This is number one. Secondly, that you can reveal that how much of the research have been done so far, and what are the opportunities that have been still prevailing in that certain area. Next part will be the methodology insights, where you can find that what will be the research designs and the techniques that we are going to be used, what these similar topics or the previous topics they have applied, and now what are the new areas which I need to go on to have new findings in my field and to demonstrate my credibility and my research in the particular area. So now I would love to tell you that there are a lot of people, okay, in this world, in this universe, I can say, they've been doing the literature, but there are some of the issues they generally confront. Let be the I can say some decisions, let it be the guidelines, let it be the biasness, let it be any theoretical and practice, okay, let it be any other issues that can be considered as a contradictory or as a limitation. So therefore, here today we will be seeing that how these limitations and these contradictions can be overcome, and you can evolve your own idea and your better understanding for the topic. And this will be helpful for the students also, those who have been also been participated from the institute, as well as the early career researchers, so that they can know that to guide any student to I can say to conduct their own research and to develop their own team. So this will provide an expertise to them in the field of systematic literature review. So we will start with that. Before starting with the systematic review, okay, I would love to tell that there are a lot of other ways of reviews. Okay, the review generally you write, okay, whenever you're going to join, just I have a question that are you people comfortable in English, or shall I use English, Hindi, or any other language? Are you comfortable?
English, comfortable.
Complete English. Okay, fine, fine. Okay, that's one. So now, so before going to any, okay, before directly jumping to the systematic review and going through its parts and the compositions and the constitutions, so let's focus on what are the different type of reviews. So for example, whenever you people have joined any group, okay, for any PhDs or MPharm, your guide have, or supervisor have definitely told you to go for a review literature. And what you did is you put a lot of papers from different databases, and simply you wrote as etal. this et al. Here has reported this has reported this has reported that. Okay, this is basically a traditional and a narrative review. Okay, this is not a systematic approach. So in the first part, you will see that this is a very traditional way. Generally, we used to write any editorials, even in the mini reviews, as well as the book chapters. So here we can see that there are, I can say it is not very specific. Yeah, but for some of the areas such as the medicinal chemistry, so if you could see that there are a lot of papers could be published with this approach. Okay, because you need to have a lot of data, you need to have a broad overview of the topic so that you can predict the SAR. So this is more related to pharmacy people. I just want to say the medicinal chemistry people, they can correlate very well that they write generally a narrative review with one of the scaffold, or I can even say that in pharmacognosy, generally if you take a plant and you just try to provide the all of the overview regarding its therapeutics, regarding its targets, regarding its everything. So here you can find that this is basically a traditional review, whereas in this systematic review we used to provide answers to a specific research question. Okay, and we have a methodology. Generally, if you contrast the methodology, okay, it was based on inclusivity of the author's perspective. The authors want to include, okay, he is including. If you want to exclude, he is excluding. But no, here you need to have that certain criterias and the predefined criterias to be for the inclusion as well as the exclusion, so that you can have a better decision based on the evidence and not simply on your I can say vague hypothesis, or based on your own will. Okay, there should not be any will, I can say. Okay, so that is the only difference between a narrative and the traditional and the systematic review. So you can also find that there are meta-analysis where you can use the statistical pool results. Okay, then you can have a scoping review where you can have some key concepts and the evidence, and it's not so, I can say it's not about the quality assessment, you are simply writing a thematic issue. So this is known as a scoping review. So you might have seen in Bentham Science, they have already given a lot of scoping reviews where a particular area you can write your article of your choice. If you are aligning with that topic, you can definitely find I can say a position in one of the special issues or on the one of the scope issues. Okay, so there are other parts which are known as the rapid reviews. Okay, so it is for the, I can say there are some editorials also. Okay, you can find, you can see the example that COVID-19 vaccine efficacy. So these are small articles which are used for awareness. Okay, so therefore the reviews have a different background, different utility. Okay, and whatever the things we are incorporating, so the constitution of that review also differs. So therefore, you can see if you're moving more towards more reviews, you can see that there are umbrella reviews, there are living reviews, integrated reviews, and one part is very important, that is the critical review. There are specific journals, okay, such as critical review in drug delivery. Okay, here you can go for a huge focus on the methodology. Okay, you need to evaluate all the strengths, you need to reduce all the biasness, you need to identify that which is the most important part, or you can say these are very focused. For example, just you do a DOE, that is design of experiment, you are taking a small I can say small area, okay, a very thin area, that whatever the factors that are influencing. So here you can find that a very critically discussion will be provided. Okay, where you can find a lot of emphasis and focus have been provided to the very thin area based on different headings and subheadings which are representing the different parameters which are influencing it. So here you can find that the critical review, systematic review, narrative review, they have a very different background. So therefore, if your supervisor is telling you to write a complete review, you need to be very focused that you are addressing that research problem that you are going to deal in your masters as well as in your PhDs. So therefore, it is very important to know that which type of review you want to start. Okay, so so you can see that these are some of my papers that I have already published. So here you can see this is, I can say, a very classical or a traditional literature review approach, where I just like you quoted the example of the medicinal chemistry. So here you can see that I have only given whatever the structural perspective, what is the molecular target perspective, which is the core area. Just you can see that Chalgos has anti-infective. Anti-infective is a huge area. Okay, it's a huge area. I have only focused on the structures and the molecular target. I have made it little small, but this is not a systematic approach. This is basically a narrative approach. These all articles. Okay, but here in you can see the expert opinion on therapeutic patents. You can see the topmost right. Okay, so here you can find that this is not that approach. This is a critical review approach. Okay, so now you can differ from this article. Okay, first of all, I've given you a left-hand side of the life science. This is a narrative review. Right side, you can have a Taylor and Francis expert opinion. That is your critical review. And you can see that I have just now given you a scoping review, and I can say a special issue based on a special talk or on a hot topic. You can see that there is a lot of difference. Okay, so topmost it is narrative, right side it is critical, and the in the bottom you can find it as a scoping review. So you can find all the difference between all the three, although I can say that all of them are providing you an information regarding chalcones in all the areas, but their approach remain certainly different. So this is somewhere you can find that they are overlapping. A question might be coming in your mind that what's basic the difference? What's the major thing that we have achieved from these three different type of reviews? See, basically the methodology will remain same. Okay, methodology will remain same in case of the narrative review as well as the critical review as well as the scoping review. Here the methodology will remain same now, but in case of the systematic review, then the methodology and the way will be very different. Okay, so next. So this is now we are talking about a broad. Okay, now till now I have talked about broad things that have been existing. Now I will narrow down to only two comparison, that is the narrative review and the systematic review. Okay, just compare between both. Okay, so we can see that if you talk about the purpose, it's having a very broad overview, whereas the systematic review will provide a very specific question. For example, I can give you an example that whether this FDP will give lot of information to all the participant or not. Okay, this is a systematic review type of a question. Okay, I need to know about a very specific question, that whether you are getting a benefit from this FDP or not. Okay, whereas if I talk about a narrative review, you will write about all the events, happenings, everything that have happened in this FDP, participants to everything. Okay, so now you can find the lot of difference between it. Now in the narrative part, we are only dealing with that what are the events and happenings. Now here in the systematic review, we are dealing with what? We are dealing with a specific question, that whether the benefit has been provided or not. So therefore, in this part, you will not get any, I can say, you will not get any, I can say, pre-formulated methodology, or I can say any protocol, or even any formal things you need to do. It's based on the selection criteria of the person who is doing that review. Okay, but here we need to have a Prisma guideline. Okay, this Prisma guideline I will discuss later. Okay, so now these search strategies will be very different. Now you can use the database, Google Scholar, Scopus, or many others that you can go through. But here databases are multiple, but you can have to use Cochrane databases. You need to use I can say science-based databases can be used. Okay, PubMed is one of the best way to get. So there are multiple databases, but I can say that there will be in the reproducibility search, you can find that it is quite restricted. So now in the next part, you will see that you can see that what about the biasness. Okay, in the biasness is very high. Okay, I can do whatever I want. I can eliminate 10 papers if I wish. Okay, to be very honest, I don't want to go for that. But yeah, if I'm going to compare with the critical review, see, you can see this side, there's a critical review. Here there was a what it is about patent review. Okay, it is, you can see that updated patent review, critical review. This comes under critical review. In critical review also, it is closely matching with the systematic review. Okay, but not with the narrative review. I don't have that choice of eliminating the patents. I cannot eliminate. So now you can again see from 2014 to present. Okay, 2014 to present. It is of the next 5 years, 2014 to 2019. Okay, getting my point? So that means in 5 years, I have to search all those patents that have been provided in the database, such as Google Scholar or the WIPO. Okay, I don't have any other choice of creating any biasness, that I cannot remove any references of my choice or patent of my choice. But here in the narrative, I can choose. Okay, I don't want this paper. This paper, if I add, okay, there are a lot of issue. It is closely matching. Okay, let it remove. No, this is not possible in the systematic review, because you need to have this as a minimized form, as well as in according to the predefined criteria. You need to have a dual screening, that whether you are able to add all those paper in the provided duration. Provided duration means whether it is a 5 year, 10 year, 20 year, whatever it is, whatever, even for 2 years and 3 years, people have conducted this kind of studies. Okay, so one of the most important parameter is the publication part. Okay, and publication requires quality. Okay, so therefore you can find that majority of the systematic reviews, okay, they generally, they come under good publishers. And you can see the medical journals, top medical journal, New England Journal, to many other, I can say, medical hypothesis, to everywhere. Okay, they publish with high impact of these studies. However, in the narrative, you can find that there are a lot of diversity. Okay, some of the paper are coming in zero impact factor paid journal of Scopus, whereas some are getting indexed into RSC, Elsevier, I can say Springer, to impact factor 2 to 10. Okay, so, or even I can say impact factor somewhere, if it is so good, then it will come to up to 20 impact factors. So here the quality remains very variable in both the cases. And that is the reason the publication part remains a big challenge. So now if you talk about a data synthesis, okay, between both, you can find that no, there is no formal analysis that you need to do. Okay, there is no need for any statistical analysis. You simply I can say in whatever you can say in Hindi, there is some alankar. Okay, in one of the alankar, it is, I don't know the name. It is that whatever things the I can see the Kavi, or I can see the poet have seen, he has represented that. It's just like Kavi ne ye dekha, Kavi ne ye likha. It is simply that. Your narrative review is just like that. You, whatever researchers have told, you have simply copied it. Nowadays, you have a very good advantage, that is Quillbot, that you use for the paraphrasing, so that it will not come under plagiarism. And simply we are presenting the same thing whatever the researcher is actually telling. But here, no, here you need to have a very different way. You need to structure your own things, you need to narrate your own things, you need to tell a new story, you have to have your statistical, okay, things very correct. You need to do a meta-analysis, you need to do your meta synthesis also, you need to have all the aspects to be very, very correct. So therefore, if you see the reproducibility, it becomes very low for a narrative review. But for the systematic review, it becomes very, very high. Now you can see that if you talk about that where my work is being cited, because nowadays citation is one of the most important part, that you have already seen, that it will affect your H index, it will affect your I10 index. Therefore, the if you talk about the audience, okay, it is very, very restricted. I wrote on plant pharmacognosy, people will read. There will be, or even I can say there will be generalized reading without much citation. But if you could see the systematic reviews, okay, it is being used by the researchers, by the policymakers, as well as the evidence-based decisions. Why I'm talking about this? There are a lot of bodies such as ICMR, there are a lot of bodies such as the government bodies which are dealing with the health. there are health departments, there are statistical departments in Delhi, in the national capital. So they used to go for this type of work for the systematic reviews. So they used to see that what is the current perspective, how the people, how the data is coming. For example, in malaria, where you can say there are a lot of data on the poverty, there are a lot of many other current burning issues across the nation. So from that, they can provide an overview that whatever the allocation of the budget, whatever the allocation of the studies, okay, can, so they can, these reviews can provide you more insights, as well as they can help you in the decision making, where to allot more resources, where to allot less resources. So therefore, traditional review, if you compare, it will not provide you that decision making, because if you see, if you go towards a progressive time, as the time goes, you can see that those papers are not being cited. Okay, that's why even if you could submit a paper to any journal, they will definitely say, just provide us the literature within 5 years. Don't provide old literature, don't provide old citations. This is the reason. But in systematic review, there is no issue. You need to just define a timeline. I can go for 50 year back, no issue. Okay, I will add a paper that was published 50 years back, doesn't matter. Okay, because I have already provided the inclusion criteria. In the inclusion criteria, I have already said that I will add the 50 years. Okay, but here now you can see the final thing, that is the example. So your editorial reviews, your book chapters, your articles, okay, this will, but here it will be very different. This will be a Cochrane review or a Prisma compliant meta-analysis, where you can find that the data is more trusted as compared to any traditional review. Now, to be very honest, that what basically is a literature review. Okay, so now I have, first of all, I have told you more about the type of review. Then I have narrowed down to, with your narrative review that you do whenever your supervisor says, write a review data, then we will start a research. Okay, you just simply go download some of the material, even from Google Scholar, and any other, then you formulate it. Now I'm narrowing down to the systematic literature review. Okay, here now you will see that this is a very rigorous protocol driven method. Okay, where there will be specific research questions, there will be one hypothesis that you need to go for. Okay, if you talk about what basically is the goal, we want that there should be reproducible conclusion, and a person who is reading this can get a decision about it, whether it is correct, whether it is incorrect, whether it requires reform, whether it require any other evidences, whether can any budgets be allocated, whether any other perspective can be defined. So everything can be provided from a systematic literature review. Now you can see that what are the compositions. Okay, basically it will always follow Prisma guidelines. Okay, guidelines we need to go for when there will be a protocol. I will tell you that how these guidelines are need to be matched with that, the protocol that you want to go for. Okay, so now I will tell you about PICO and PICO. Okay, PICO and PICO, that is the formulation of the research question. Okay, formulation of a research question is the most important part for you. That because somewhere you need to justify your own thesis, your topics, your areas. Okay, your area of interest and your research expertise all have to be aligned to that research question. And here one thing I just want to tell you, that there will be a quality assessment. There is a Cochrane RB tool, or we can say ROB tool. Okay, it's already being used for the quality assessment. So there are, for example, I can go for a questionnaire based study also. Okay, now in a questionnaire based study, I can say, what is your name, how you're doing, how are you fine? These are not the question. This should not be the question. It should be very specific. If I want to know about the wellness, I will talk about whether he's taking the breakfast, whether he's taking multivitamins, whether he's suffering from any diseases. So these should be the questions. Okay, this will define the parameter. Just like you have a NAC, you have an NBA. Okay, there are certain questions that you need to go for to define the quality of your institution, that whether those quality parameters you are following or not, to maintain a certain quality for your institution. So therefore, for this study, You need to have that quality, desired quality to be incorporated, and that through the Cochrane ROB tool can be used. So now, if you talk about these steps, that whatever the consecutive steps that you need to take, first of all, you need to formulate a question. Okay, I can say whether from pollution, okay, can pollution cause diabetes? Maybe, may not be. Okay, it is a hypothesis. This is a question that I've given. Okay, now I can give you what is the protocol. I need to take the pollutants. I need to take, I can say, I need to define that what are the pollutants that are being circulating in the air. Okay, that we are inhaling. So for how much duration we are inhaling, for how much, how many people are inhaling in a particular area, everything has to be very, very defined. Okay, so if you want to go for the literature first, okay, related to this area, I need to prove that, yeah, this is correct. Okay, I need to go for a literature. Okay, from literature, I can prove. I need to go for databases. I need to go for Cochrane. I need to go for PubMed. I need to go for many other databases. Now, how I can screen the studies? See, there will be some inclusion criteria. For example, a person who is suffering from lung cancer, okay, should be excluded. I am not going to include that person. Why? Because he will be on nebulization. You know about nebulization. He will be having the things nebulized. Okay, he's not getting that fresh air every day. He's not moving here and there. So these patients must be excluded. So from the Prisma flow diagram, I can say that where to exclude, where to include. Whom to include? People who are going to office every day, just like your teachers, your students. Okay, you are coming every day to college and going back home. So this to and fro. So your exposure duration, your everything, these need to study. This will come under the inclusion criteria. Okay, so now we need to extract the data based on the databases. We need to synthesize that whether we want to make it as a more narrative or more as a statistical based. Okay, we need to have this option, but we need to include all those areas, all those I can say duration, okay, from where we need to study. And that is the only reason that we have this meta analysis, that is a statistical part, to be correlated with all those paper which are being included in the study. So now at last we will get the report finding. We can provide you a good report. Okay. So now there will be such strategy. Now you can see there are many papers nowadays. I am seeing is a blend of narrative as well as systematic review. Okay. Why I'm telling you this? I have seen paper, many reviews published in MDPI, published in Springer. Now they have, after introduction, now they are writing as a second point, inclusion criteria. Okay, what are the paper that we have included? We have included this topic. For example, is work on diabetes. He will write diabetes, comma, and slash or. Okay, diabetes or anti-diabetic and slash or. Okay, like this. Okay, these are the, I can say these are known as the search strategy. Which keywords you need to add, how you can filter those paper from the papers which are only having keywords. See, if I put a keyword as chalcone, antimicrobial, and a target, okay, if I go to Google Scholar, you will see after 15 pages, Google Scholar will provide you only those paper where there is a keyword either as a chalcone, either as antimicrobial, or either as a target. Okay. After that, you will find that all papers are not related to each other. So this needs to be excluded. Okay. So this process is known as filtering. Okay. Documentation filtering. Okay. So therefore, you can have your search strategy for the different databases, and you can go with your most appropriate keyword for your search. So now, if I talk about the inclusion criteria, this is the most important part, because you need to conclude something. Okay, you need to conclude based and you have to prove your hypothesis. Okay, so that you need to have a time duration, how much I need to add, which study population I need to add. Okay, for example, in Haldia there is a huge port. You already know about a huge port. Okay, how many ships are coming, from which country they are coming, okay, what they are taking, Okay. Likewise, so I can give you example that what are the types, what are the population, what will be the outcome, so why we are doing it. So these are certain things that we need to address in this systematic review. Now you can already told you about the quality assessment. I've already told you about this is one of the Newcastle Ottawa, okay, protocol, that for the observational studies, that how for example, I want to conduct a study on TB. I went for a to any limited area, okay, or I just want to go for that whether he's taking the dots or not, okay, just like an observational study. I'm not going for any approach, a deep approach where I'm talking about the doses, I'm talking about this and any other I can say very specific informations. So in the observation study, we need to add Newcastle Ottawa protocol for the quality assessment. Now the synthesis method, this I will teach you later. The outputs, I can say it will be first of all there will be a Prisma flow diagram. So we need to see that whether we have added all the type of papers in the limited duration or not, and we have excluded those criteria that which are not fulfilling our demand. Okay, whenever you are going to add any content from the exclusion criteria to inclusion criteria, your statistical data will vary. Once statistical data will is going to vary, your conclusion will be a very vague. So therefore, just say there is one we can say a proverb that one rotten apple can spoil the whole goods. Okay, so therefore only one exclusion, improper exclusion criteria to be included in inclusion can destroy your whole study. So therefore you need to be very very specific. Okay, just like you told, जैसे you say ना कि सड़ा हुआ आम ठीक है सारे आम खराब कर देता है. So don't do those kind of mistakes while you are going for a systematic review. Okay, now you need to have the evidence based tables, that whatever the things that you have got, what you have observed, what you have analyzed, when you have statistically or non statistically analyzed both, as well as there are a lot of plots related to, you can see the forest plot, or if it is a meta analysis. Okay, so here you will find a lot of advantages. So there will be no biasness. It's nothing but biasness. From narrative review you cannot conclude that that whether the allergens in air are causing diabetes or not. Okay, until unless there is a molecular biology based evidence, okay, that it is for example, if it is going for your PPAR or PTB1B or DPP4 or like this type of targets. Okay, until and unless it is proved through your molecular biology techniques, such as I can say Western blot or ELISA or any other ways. Okay, so here you can see that from that data you can have a very credible source that you can have. You can trust this data for your decision making, so that further researches can be done, okay, in the upcoming time. So you need to enrich your own area. So this is one of the I can say very best approach that you can use in pharmaceutical sciences, because majority of the pharmacy people are into two areas: narrative review and critical review. Okay, so just you need to shift from and whereas the medical people are more focused towards the systematic review. Okay, because they don't have much time, okay, going for more and more literature, so they always prefer that there should be systematic review where I can take a decision for the therapeutics, because they are more into patient. Okay, they're into more into patient care and all. So therefore, the therapeutics are more addressed through systematic reviews. So you can see that there is a limitation also. So every good thing has a limitation. Okay, nothing like I'm providing all the good things. Okay, there will be limitation in each and every. There will be pros and cons. So if you talk about cons, okay, there is this is quite time intensive, because it will take a lot of time. You need to go for all literature. You need to go all databases. Okay, Cochrane, PubMed, Science Direct. There are others. Okay, there are other medical databases that you need to go for. So you require expertise, because you need to know about ANOVA, you need to know about Prisma, you need to have about forest plot, you need to know about meta-analysis, meta-synthesis, you need to know many other things. Okay, so that means an expert can do it. Okay, a newcomer will find it very, very difficult to do it. But if he can do it with his own expertise and the support of the guide, okay, those papers will reach new heights and new citation limits. Okay, you can see that there will be unlimited citations to this type of studies. Okay, and somewhere you can find that the existing study quality is also limited. So whether to include, whether to exclude. Okay, you talk about one word, there is no dharm sankat. Okay, add karu nahi karu, to add, exclude, what to do. Okay, somewhere this problem comes due to the quality. Nowadays there are a lot of journals which are paid, so many people this is formulate using AI. Even your, if you talk about uh, writing this paper through DeepSeek. Okay, DeepSeek is one of the I can say just next to chat GPT. DeepSeek can even provide you systematic review, okay, on any topic you can ask for. So these generally reduces the credibility of majority of the studies. So now you can see some of the examples such as Cochrane, okay, reviews, and WHO guideline reports. So these two examples, okay, if you are a student, do remember these two examples. This is very important because Cochrane is the standardized I can say a database where you can definitely go and can perform systematic review based on various topics of your own choice. Okay, let it be therapeutics, let it be related to lifestyle, let it be on nutraceutical, let it be anything. Okay, just you need to formulate a question. Now, what are the key steps in conducting the systematic literature review? So now you can see that there is a paper that I'm shredding that paper. Okay, now this is the whole knowledge people are shredding. So what it is representing, that different people, okay, different people have provided different area. Now I can tell you a very good example. The many people say academia join karo ki and industry join karo. You know, in pharmacy, both are very different. Okay, both are very, very different. Just like a pyramid. Industry is a pyramid. All people are going for one goal, one drug. They want to manufacture, they want to market, they want to take patent, they want to take intellectual property, they want to modify it, they want to do better. Okay. Whereas in academia, okay, this is a reverse pyramid because you start from one and you are diversifying. For example, I can tell you in your institute, you have a lot of teachers, a lot of different approach, a lot of different specialization, a lot of different research interest, and therefore a lot of different kind of papers. So here you can see different people with different ideas. Okay, it is floating. So you need to provide, okay, the systematic review approach where you can make it as a triangle. Okay, you need to make it as a triangle. Okay, now you can see there is a triangle, that is the systematic review. Okay, so you can just see the steps now. Okay, I will discuss that triangle. Okay, that triangle is very important. Okay, why I'm focusing on the triangle, I will explain in the next slide. Okay, now you can see the steps in a systematic review. First, you need to have your topic selection. That is pretty important. Okay, then you have to conceptualize and create a protocol. That, for example, I give you a very good example that whether this allergens can cause diabetes or not. Okay, you need to eat. This is a new concept. This is my concept. Okay, this is my hypothesis. Okay, I can go for. I can create a protocol of how I can conduct this study. Okay, now. Which type of database I need to use. I'm searching for the studies. Okay, whatever the things that are already being there. Okay, so I will go for a screening based. Okay, which papers I need to include, which papers I need to exclude. Okay, after the searching for all the studies. Okay, now first of all, what I search from PubMed, search from many other this. Now I will see that whether I need to include, I need to exclude. Okay, then the next will be that how I can appraise a study. Okay, how these studies can be reframed, how these studies can be, I can say, I can formulate, I can go for a metasynthesis, that okay, these are the key areas which are pointing towards them. Okay, now next will be the abstract data. Okay, I need to take the abstract of those parts, which are the most important thing which are being given in the paper. You have, see, there are a lot of in a paper, there are a lot of different aspects are being shown. Nowadays, what I'm seeing in pharmacy, that nowadays what people are doing is they are simply taking a national product. They are simply doing activity like anything, anti-diabetic, anti-inflammatory, antioxidant, whatever the things they're given. Okay. I need not to read all those aspects. I just need to extract that abstract part. Okay. That how the people have studied, how they have conducted this. I just want to take a gist. I will just simply take the abstract part. Okay. I'm not going to take a lot of things from the whole paper. I cannot read 700, 800 papers a day. Okay, or in a 2 days or 3 days. Impossible for me. Impossible anyone. 700, 800 papers to read. Therefore, we need to go for the abstract. Okay. From that abstract, you need to synthesize that what are the things that remains very common. Okay. So yeah, these 5 papers are common. 7 papers are common to this aspect. 9 papers are common to criteria number 3 aspect. Likewise, you need to frame the criteria. Then you need to interpret the results of it, that yeah, whether it is matching those criterias or not. So this is the way you need to extract that results from those published papers. So here now we are dealing with, okay, all the research papers. So keep in mind, nowadays what many of the researchers do, or I can say newcomers do, they are writing review from a review. Okay, I have seen they are taking 15-20 reviews, okay, copying it, putting in quillboard, paraphrasing it, done. At least I can say guide ko khush karne ke liye, to make the guide feel very happy that the boy has done something very good overnight. Ek raat baithe aur he has done a lot of good things. No, it's not the way. Okay, here in the systematic review, you will only focus on the research aspects. We are only dealing with the research aspects. We are not doing that malpractices. That malpractices can be stopped completely, okay, through this systematic approach. Now here I can tell you that what is the logical consistency and the synergy across all the steps. Okay, now you can see that first of all you need to formulate a problem. Okay, just like I said that there is a topic selection. Okay, first, what is the problem that is existing? Just like your diabetes. Diabetes is a huge problem. Okay, it's a huge problem. Okay, next, what is given in the literature? Can I go for the screening of those most potent papers that have been already being there? Can I go for the data extraction and the critical appraisals? Then can I go for the analysis and meta-analysis? This is a statistical part. Then I can go for the results. I can say dissemination is nothing but whatever the results and the conclusion. And whether I am seventh part is most important. Okay. Nowadays, this is one of the criteria for the interview. I can also say that whether your work is of worthy or not. Okay. So whether your work, whatever the work you have done so far, okay, it is of any worth to the scientific community or not, whether you're updating any or reanalyzing any data, you're providing a new area, you're providing something new, whether yes or no. So these are certain things which you need to focus on. Now this was the pyramid. Okay, the pyramid which I was talking about, this is basically the pyramid. So now you can see that whether and where these things are lying. Okay, why systematic review is considered as the best? जैसे बोलते हैं ना, राजा consider करते हैं, एकदम राजा, royal consider क्यों करते हैं? Why it is considered as the most royal part? Okay, because you can see the bottom of the pyramid. What you are doing in your laboratory is an in vitro research that you are doing it. Okay, just to have your final year projects being done, or I can say some of the MFARM projects also were employing I can say 75% in vitro and 25% animal, or even I can say with no animals also I have seen. Okay, such as the cell line studies and many others. So this will come as the bottom of the pyramid. Okay, for you have done a huge research. Okay, for people who are into decision making, okay, therapeutics, government positions, For them, this is nothing. So sometimes you might get frustrated that you have done a lot of work and you are not getting a very good recognition. So this, so you can see where you are lying in the pyramid. Where you are lying in the pyramid.
Kindly, everyone, please mute yourself.
Yeah, please mute.
Kindly,
Yeah.
everyone, please mute yourself.
So please mute. Yeah, thank you. So now you can see that. Yeah, now you can see that you might have done a small work or a good work. Okay. But now where you are lying in a pyramid? Your work is simply you are providing a torch light in a dark room. Okay. You are simply providing a torch light in a dark room. You are simply providing a path. You are exploring a path. Okay. Whether, for example, whether XYZ plant is having, okay, anti-inflammatory activity or not, you are studying it. Okay. Either through in vitro protocol, either through in vivo protocol. Okay. Simply you are exploring it. That's it. Nothing else. Now, when it will be composed in an idea or any opinion or in an editorial, then your work will be highlighted. Okay. Your work will be highlighted at that area. Then there will be certain studies based on, I can say, patient. For example, you have taken XYZ plant and this plant is showing anti-inflammatory activity. Now, some of the, I can say, very knowledgeable people or people at good positions, they might write about this plant. Okay. Now, some of the people will find that can we do a case study of it? The Ayurvedic people are doing it. Okay. They are doing a trial based, or they are using a case study, or they are using a case control studies. Okay, so they will be go for a case study. They will have their own methodology. They have their own way of dealing with the patient. Okay, so they will represent a case report. When there will be multiple case report, it is known as a multiple case study. Okay, so lot of means at least 100 people, or I can say 50 people. That means a significant amount of, I can say, significant amount of population have been studied. Then there will be case control study, and here you can see the approach for the first step of the clinical trials. Now, this case control study, cohort study, and the clinical research, okay, these all are coming under an important part. Okay, now after the confirmation, now we are going for the formulation of any formulation development or any kind of uh, dose delivery, or I can say any other type of ways, approaches, so that we can study in a larger population. From that data, okay, from that data comes the systematic review. Now, sitting at the top of the pyramid, now we will be studying from bottom, that how these hypothesis or how this study have been formulated. This is studied from an in vitro model. Now this has went through the trials. Okay, it's known as the randomized clinical trials. So now after the trials, after the confirmation, then comes your systematic review approaches. So now you can see in the right that from the question, okay, you can first have a search, that whether I need to have a search somewhere. While doing this search, you can find that there are some issues. For example, your guide might say, इसके ऊपर थोड़ा search करके देखो. You just go to this, okay, and search something. Okay. You will say, sir, I only got 20 paper. There is no more paper available. Okay. In narrative review, it's okay. In narrative review, it's okay. That means not much work have been done. Okay. But in systematic review, things are very different. Okay, you need to first of all think that, okay, whether it's okay or not. If I go ahead with that 20 papers, it's okay. And I will try that I can include at least 100 papers, because since many people from the chemistry background, even they could understand, or even in the pharmaceutics or background, that the number of samples, a minimum number of samples are always being required. Okay, but people who are doing QSAR, okay, they want that at least the IC50 value of at least for the test and training. Okay, this should be at least more than 40 to get a best score. Okay. Nobody does a QSAR with 10 compound. The number of samples must be bigger as to become statistically relevant. So therefore, the question can be modified. It can be broadened up to include more paper. Okay. For example, I told allergen can cause diabetes. Okay. Now I will say what? Allergen as well as food contaminants. Okay, from food. Now what I was telling about, I was telling about air inhalation. Okay, I was earlier talking about inhalation. Now I'm talking about from food. That means I am introducing allergen, okay, antigen from both the sources. Now my paper will be increased. Okay, so that's why it is given as the modify the question. Okay, now write the, then if it is going well, then just write the protocol that how you're going to do this study. You need to register first. Okay, for any of the studies, you need to first of all you need to register. Then you need to go for the searching, screening, extraction, meta-analysis, reports, and then we can go for either. See, in the data extraction, there will be summarizing, that is non-statistical part. Okay, and there will be meta synthesis where there it will be more about the more and more statistics part. Okay, so both the approaches can be utilized, okay, here. So now, but majority of the thing I can say, whenever there is a meta analysis, okay, statistical inputs are mandatory, and that is the reason the reports which we will present after this systematic review will be very trusted. So now, and therefore, whenever you are providing it as an update, many researchers will start working in that area. So what after this update, you can see that again there is searching. There is again searching. So what happened? For example, when I did my M.Pharm, okay, for example, the paper that I published was during my masters. Okay, that was in 2015. At that time, in my masters. Now it's a 10 years long journey. It's 2025. So people, if I would have published it as a systematic review, what they have done, they would have taken that data as a fixed data. Okay. And after 2015, 16, 17, 18 to 2025, they will go to that journey, thereby adding the paper on the existing data, just like you construct your buildings. First of all, there will be ground, then there will be first, then second floor, third floor, fourth floor, just like. So we never ever demolish the ground floor. So this is the thing that we are simply updating. If the question remains same, okay, we can go for the update and search, update and search, update and search. So you can now I think you have got a very good perspective. Now I would love to tell you that how you need to do. Okay, this is what I can say. This is a protocol I have given. Now, how you will do it? So first of all, you need to assemble a team. This is very important. Okay, you need to assemble a team. That is pretty important part. You cannot do everything at your own. If you are going for a large sample, if you are going for a large study, okay, it is merely impossible for you. You will get confused. At least you need 3 to 4 person having different expertise of data collection, of continuous literature extraction. A biostatistics person is very important here. Okay, because many I can say I can frankly say that pharmacy people don't have that level of biostatistics knowledge. Yeah, it's true. I even don't have that level of knowledge. Nowadays we have AI tools. So therefore we are using AI tools to get that which formula to be applied, how much to be applied, where to apply, when to apply. Okay. Earlier it was a huge challenge. So now next will be to develop a question. Just like I've given you example, the inclusion exclusion criteria protocol, that okay, these things we will do. Just like you have a timetable, you have a schedule. Okay, you have a schedule every day, that this is first class, second class, third class, then there will be practical, there will be library time, there will be this period and that period. Okay, like that. There should be a protocol that after that what you will do. You need to have a search strategy. You need to have You need to go about which databases you want to go, whether you want to go for the documents, whether, okay, we need to go for those research documents, how you will manage those documents, that is pretty important. Okay, because there will be a lot of things, how you can segregate them, this is one of the challenges. You need to go for those screening, that after segregation also you need to screen that this is for this period, this is for that period, this for that. Then you have to include those study, then you have to extract, analyze and synthesize the data, and then you only can write and publish this manuscripts. So this is what you need to do. Earlier things are, see, we can revise once. Okay, we can revise. Okay, one thing we can revise, that in this part what we have done is how the systematic review is generally being done. Okay, we have given a path, and here where we are standing, if you are doing a systematic review, where we are standing in the scientific community, we are standing at the top. Okay, so how it is being done in a generalized way, I have given in the question to update in the right hand side, and how you will do is you need to do from these 12 steps. Okay, so keep in mind that this is very important part, where you will be acting not as a researcher, not only as a researcher, but as a decision, as a fundamental person for a decision maker, for decision making, I can say, motivation. You will motivate others to make a decision, okay, based on your studies and based on your results. So therefore this is the importance. Now we have been discussing about the PICO and PICO. Okay, so PICO framework. Okay, now you can see one of the question. First of all, you need to see that in a patient of type 2 diabetes. Okay, so here the P is referring to the patient. What kind of patient or what kind of population it is? It is describing the group. Okay, this is known as the PICO study. This is very common in the clinical research as well as the medical part. Even in pharmacy, people are now being using it. Okay, so this P referred to as the population or the patient. Now you can see what is that population. This is the type 2 diabetes. Now metformin therapy. Therapy means intervention. You're intervening. How you're intervening it? You are intervening through giving a new drug. Okay. What is C? C is a comparison. Comparison with what? With a placebo. Okay. I know very well that you know about the placebo. I need not to tell about what is a placebo. Okay, this inactive substance. Okay, so and how is the outcome? What is our outcome? What we want? That we have to lead the whether there is a reduction in HB1AC level. HB1AC level is one of the level that has been responsible for that is the in diabetes there is a enhancement in the HbA1c level. Okay, this is one of, I can say, one of the most marker that whether to confirm that there is a type 2 diabetes or not. Okay, so here, so you can see this is known as the PICO. Okay, PICO framework. You need to go for PICO. Earlier in is now compare with narrative. Whether we are using any PICO? No, we are simply narrating it. We are telling a story. Here we are not telling a story. We are only providing a direction that, yeah, this is the way, this is a problem, whether this problem is existing in society or not. If it is existing, we need to provide a direction. This should be the approach. Okay, next, there is a PECO approach. Okay, PECO. Generally, when there is intervention is not applicable. Okay, there is no intervention is applicable. Earlier what I've given, metformin. Okay, metformin is a drug that we are deliberately giving. Therefore, we are saying that it is an intervention from our side. We want to know whether metformin is going to work or not. Now in the PECO, you can see that is the exposure. This exposure is not desirable. This is undesirable. A person who is going to somewhere XYZ place, he is going to mines, he's getting in contact with methane. Okay, is it desirable? Do we want that? We want to feed methane to that person? No. Okay, but he's exposed to methane. This is undesirable factor. Just like you have in your formulation optimization, there are certain factors independent and dependent. Just like we have also have this exposure as one of the factors. Okay, here you can see do urban resident. Now see urban resident. What is urban resident? It is a population. Okay, it is a population. This is very important that whether we are studying to which group, this is known as the urban resident. Now, uh, is the exposure? Exposure is what? To the air pollution. We are also providing long term. Long term means what? That we are also defining the duration. Okay. As compared to in low population area. For example, I'm comparing this with the A grade city and the C grade city, A tier city and C tier city. Okay. We can compare. That is, C will be comparison. And what is the incidence of the respiratory disease? That's why in Delhi, okay, in Delhi, a lot of respiratory diseases are there. But I can say in Haldia, it will be very low. Obviously, it will be very low. Okay, because of population, because of the industries, because of a lot of things. So there will be reduced incidence of the respiratory diseases. So therefore, in this, with this study, okay, so the major difference between PICO and PICO is intervention and exposure. One is deliberate, one is non-deliberate. Okay, you need to know about this. Now, why we need to use this PICO and the PICO? Okay, because we need to focus the research question. Okay, we need to see that which database we need to use. For example, if I'm going for a respiratory based, okay, then I have to go for respiratory databases. Because if you want to go for a diabetes, I need to go for the diabetes database. Okay, so we need to go for it will guide you that which database to go for. So this will provide you more reproducibility, and it will improve your inclusion and exclusion criteria. You can differentiate between, okay, I want to add this, I want to add that. I want to exclude this, I want to exclude that. It will give you that confidence. Now, the major what I'm using is confidence, because the person should be very, very confident to those things. Okay. Now, you can also modify based on PICOs by adding S as the study. Okay, that whether it is, if there is a, I can say that in any therapy, whether it is addressing to certain areas, okay, or whether it is going for trial based studies. Okay, for example, you can see that the anxiety based in a college student. Okay, O is the, I can say in that point is the outcome. Okay, P is the, in the college student is the population. Okay, mindfulness therapy, that whether it's a therapy, we are doing it deliberately, that whether we are providing a therapy to those students, that whether, so these areas must be added, so we can modify the PICO. Okay, so this will give you more. So you can see PICOT, that's the time frame. Okay, 6 weeks, 10 weeks, 12 weeks, you can add. Okay, even you can formulate in a different way. This is known as the spider means sample, phenomena of interest, design, evaluation, and the research type. Okay, so these are some of, when we are going for a qualitative research, we are not going for the quantitative research. We need not to go for the PICO, we need to go for the spider. Okay, so there are some of the mistakes which generally people do, which affect the exercise. For example, what are the effects on the exercise on health? Okay, so these are people used to write as a big, okay, or I can say very unfocused, or you can say too many results. We need to narrow it down, but we need not to make it too narrow. Okay, so for example, 20 mg of a drug will reduce, okay, the blood pressure in a 50-year-old. Okay, this is also too much narrowing. We need to broaden up. Okay, you may exclude the relevant studies if you could do that. You need to have things very focused. You should not have a very generalized thing that can lead to a lot of results, or you can go for I can say a very narrowing down the question that lead to majority of your paper being excluded. Okay, so if you are for example, here I can say, I've given that does meditation reduce stress. Okay, whether it will come as a yes or a no, or whether there is intervention or there is a non-intervention, we need to control that. Okay, we cannot compare to this area. Okay, we cannot compare between yes and no to the some of the levels. So that is the reason that PICO must be very, very defined. So this is the most important part if I go for the step 1. Okay. No need. Okay, so we'll go with directly with the protocol. Okay, in the protocol, if we need to have a predefined plan that whether what is our basic objective. Okay, this is first of all need to be defined very well. Okay, then whatever this strategy we need to go, what are the keywords, what are the databases we need to go, inclusion, exclusion, data extraction, biasness will be there. So we need to reduce this biasness. Okay, narrative review, everything is very biased, but in systematic review we need to have that biasness to be minimized as low as possible. So we need to go for statistical, not statistical, mixed approach. We can also go for. So if you have a very defined protocol, reproducibility will be good. Okay, you can go for registration in many of the databases that we want to go for the study. Okay, so that the databases can provide you because the databases are now having two aspects. One is you need to register. People will know that what kind of study it is being already conducted. Okay, they are conducting, so that you are not going to reproduce the same work. Sometime it happens that you are doing the same research or you're doing the same kind of work every time. Okay, so if you could register, you can have a knowledge that whatever the previous work have been already being done. Okay, so you can choose your topic much better. Okay, so this will improve the efficacy as well as the biasness can be 100%. It will be reduced. Okay, I can say up to 100% it is possible. Now this is the Prisma guideline. Okay, what I was telling about. Prisma guideline is the most important part. Okay, now you can see that these are the evidence-based minimum set of items for reporting systematic literature reviews. Okay, so this is the minimum portion that we are requiring to have our study being done. Okay, so there is a checklist. If you go for a Prisma guideline, there are 27 criterias, but we need to go for certain things. No, we need not to study the whole part. Just like you have in your research paper, you have introduction, method, result, and discussion. These are also the part. Okay, these basically, if you can now see the structured abstract nowadays, you will see the front page. You will see this type of thing. That there will be a title. Okay, and after that, in the abstract, there will be a background. There will be objective. There will be method. There will be data synthesis. Nothing about the result. Ethics and registration is all about whatever the ethical protocol and the guidelines that you have taken. So why it was required? Okay, now the question is why it was required. Why we require a Prisma compliant protocol? Why don't we require a randomized protocol? Okay, because this will give you a very good background that why we are doing it, what earlier studies have been done, of what we want to explore, which population we want to explore, how much duration we want to explore. Okay, so all things. Okay, from your keywords to your selection, it becomes so unparalleled that you will be getting the most genuine result ever. So this is the most defined area. So now in some of the steps, I will go very fast to those steps. You can see that first of all need to define the question that already given an example. Search strategies just like Scopus, PubMed, and the Cochrane library databases. You need to go with the specific keywords. Okay. Now I can only tell about the Boolean operator means and or not, and slash or slash not. Okay. So somewhere we used to write and, somewhere used to write or. In pharmacy we feel okay, it's okay, and or we can write in a review. No, and or has a very different, just like in a probability. Okay, 1 by 6 plus 1 by 6 and 1 by 6 and 1 multiplied by 1 by 6 has a very different thing. In a probability you can see it is just like that probability. Boolean operators are based on that probability only. So now you can have a lot of things. Now you can go, if you go for a filter, you can have to go for the publication year, language, and the study type. Now, if you go for the gray literatures means which are not as a documentary evidences in your generalized database, such as preprint, Now you have a research square preprint being available. Okay, those preprints are to be trusted, not to be trusted, like that. Conference abstract, full paper is not been given. You cannot take a good decision of it. Clinicaltrial.gov. You need to go for a separate section, I can say separate website, and you need to go for this the current status, as well as you cannot directly introduce those data into your normal reviews. Now, what are the eligibility criteria? Is the inclusion, the exclusion. For example, if you go for the randomized trials, okay, randomized studies, or you can say cohort studies. Okay, you can see that there will be a population you need to take. That is the adult population you can take. You need to go for the intervention. For example, a telemedicine app. Whatever the outcome is, I've given you that example. Okay, for the anti-diabetic. Okay, what you need to exclude? We will be excluding pediatric study. We are only what we are adding only adult. Those who are above 18, those who will be below 18, all will be pediatric, teenager, adolescent, all will be excluded. Okay, so these are providing you a good data, a good insight about inclusion as well as the exclusions. Okay, so next will be the screening process. How you can going for the screening. Okay, so there will be title, there will be abstract. Okay, there will be for wherever you are getting a full text or not. First of all, we'll go for the abstract screening, that whether I'm getting the desired information or not. If you're not getting, you will get from the full text. Just you people do from I can say you open a paper, then you find that whether from the abstract I can write a review or not, because paper is not opening, not given in Sci-Hub. In Sci-Hub, you are not getting a paper. Getting a point? That was a level 1. If level 1 holds true, mission accomplished. If the level 2, if it is not a mission accomplished, that means you need to go for the level 2. That is, you need to open it and you need to go for a full text screening. Okay, how you can open? Now it's up to you. You need to have that access to everything. Now there are some tools I can tell, RAN and Covidence. Okay, these are the two tools that can be used for this screening process. So you must note down the name of these two tools. Okay. Now there will be data extraction. So we need to go for the author, year, sample, key size, everything. And what are the tools we can use? We can even use a Excel, Google Sheet, or specialized software such as Revman. Revman can provide you just as Excel, but with little AI functions. Okay, so therefore your sampling and your extraction remains much better. Now we need to do the most important part, that is the biasness assessment, so that you have the quality. So I've already given the name of these two tools before. Okay, I'm not explaining this. So data synthesis plan. Now we need to go for, if it is a quantitative data, okay, then we'll go for the meta-analysis. Then we go for the meta synthesis, or either as the narrative synthesis. If it is having a heterogeneous data, we can go for multiple domain data and we can make it as a narrative synthesis, because there are data from different origins and sources. Now, if you go for the protocol registration, we need to go. I already told you, you need to go for the registration. Registration will help you for two things. You are not repeating the same thing, as well as there the in the registration portal, you will get a lot of access for the different type of tools. So therefore, Prospero and the OSF, that is the Open Science Framework, is the most important part. Now this is the Prisma flow diagram. Okay, you need to go for. This will provide you a very good insight about how we have done the identification. We have taken a record from different databases. So how many papers we got? 1217 from Medline, 215, MBase 261. Okay, from Web of Science 255. OpenGraph we didn't use any, and the ProQuest and the Sinel we got 255 and 231 respectively. So in addition records, we got other from 40. So therefore, we found that there are the duplicates. The duplicates have been removed. Okay. Now the number of papers are 305. Okay. So after screening, we didn't find that some of the papers are matching to our expectation. It is not sometimes what happen. Uh, the searching system, they go for the keywords. So in a cancer paper, you are getting about anti-diabetic, because in the introduction part, people have written pyrimidine is showing anti-cancer, comma, anti-diabetic, comma, anti-inflammatory, comma, etc., etc. So it has already catch one word, anti-diabetic, so it has been included. So we need to exclude it. So now we need to go for the full text articles. Okay, we need to access. Earlier, during the screening process, we are only going for the abstract. Okay, we are not reading all 305 papers. It is merely impossible for anybody to do in a single day or a day or 2 or 3. Okay, because papers have a long perspective and they are very lengthy. So now you can see that 161, okay, total articles can be, okay, can be accessed. So for example, somewhere we're not getting those full text, we can exclude it. Or in some of the study, there are only qualitative synthesis. We want to exclude it. So now you can see that from 1217, okay, 1217 literatures. Now we have how many literature? How many we have going for inclusion? Only 4 studies included in the meta-analysis. Okay, only 4 out of 141 full text. Okay, that have been now we have excluded. Okay, see, first of all, I'm also repeating again. We have, first of all, there was an exclusion for the records. Okay, from that record that we found that these are not the suitable paper. Now we have 161. Okay, that has come for the access for the eligibility full text. After full text, we are showing that there are a lot of issues. So these issues are already being given. There is no socio-economic status being given. Sample size is too big. Somewhere no expenditure data is given. Somewhere it is not given any other data. So we are also excluding it. Now then we are going for a qualitative synthesis. Here we got 14 from the database and the 6 from the gray literature. Gray literature, you know it very well. Again, I'm telling you what is the gray literature. Preprint, conference abstract, and clinical trial are the gray literature. So from that, we have only included 4. So how many studies were there? 1217. How many we are using? Only 4. Now see how people will trust. Now see how people will trust. Because people will trust because they got that exact 3 data from database and 1 from the gray literature. So now the searching strategy, you know it very well. I've already given you know these databases very well. Scopus, Web of Science, Google Scholar, okay, many others. Now I can tell you the gray literature. I've already given you that clinicaltrial.gov, ProQuest dissertations, that is for the thesis, and for the conference it is IEEE or ACM. Okay. So here you need to go for the most important part, that is the keyword and the Boolean operator. Okay, Boolean operator means and, or, and not. This is the most important part because or is a very broadened term, and is a very narrow search, and not means exclusion. So these 3 are the most important part for your keywords. Therefore, earlier I've already given that what are the keywords you need to go for. Okay, see, you can see the first in the population. You can somewhere you can use T2D, type 2 diabetes. Somewhere you can use T2DM. Okay, somewhere it is intervention, you can see as a telemedicine, it is showing telehealth. Synonyms can be there, you need to use those. HbA1c, you can also go for the glycemic control. Okay, so these are the areas where you need to focus. You need to go for the more and more paper. It's not like that you are simply putting a single keyword and then you are saying that, oh, my job is done. It is impossible. You need to search in the most effective way so that no paper should be missed from you. Okay, there will be some advanced technique, just like we used to do, like for example, in the chalcone, when I was searching, I used chalcone. I didn't use chalcones. SSS, we never use S. because then it will only go for those literature which is having S. It will exclude the chalcone part. Somewhere we used to go for the truncation. Okay, truncation we used to use, and as well as we used to go for the wildcard. That wildcard means women and women. Okay, one for one women or one lady. That for more than one lady, women. So we can also use a phrase search. Phrase search means exact match. If you're putting in between these two, I can say inverted commas, you will definitely get that more better search. Now you have a search filter that from the publication, then the language, the study type, and the full text. You need to validate, you need to test search, you need to go for the iterative refinement and the peer reviewing. These are the three approaches you need to go for, and then you can document the search strategy. For making the transparency, you can make a search string that, okay, this is the syntax, such as hash PubMed, just like you do in LinkedIn. Okay, hash achievement, hash citation, hash research. Okay, just like you need to do a syntax. Now you need to know about the number of results that have been given, and you need to rely more on the plasma flow diagram for the inclusion and the exclusion studies. Now we will go for the selection, screening and the selection. That what is the purpose that we are going for the screening and the selection? It must be very, very defined. Okay, how many record, how many to remove, how many to access, how many to exclude. So these must be very, very defined. So these are the stages where we can know about the identification, screening, eligibility, and the inclusion. Okay. Now you can go for the step by step, that is the removing all the duplicates, then screening of the abstract, then the full text. That I've already been discussed. So now there will be a final conclusion, and the tools for the screen will be Ryan, Covidence, Excel sheet, and SR review. So these are the 4 tools you need to use. Now the people who have already joined a PhD or a master's, I can suggest that go for a systematic review instead of a, I can say, literature or a narrative review. You will come to know about this tool, and this tool will do work for you. So do smart work. Okay. So smart work is always preferred. Now it is the quality assessment. Now we need to know about how the biasness to be removed, how weightage to be put, how the credibility must be improved, and how the guidelines are to be complied. Okay, these are the 4 things we need to know about. Okay, to make our quality much, much better. Okay, so I can already told about the randomized control trial. You need to use the Cochrane ROB. You can use observational study, that is the NOS, non-randomized studies, that will be Robbins, and the quality study are already given in the last, that is the CASP. So this will help you in the randomization, selection, biases reduction, as well as the methodology. Now we have the Cochrane, that is the risk. Okay, risk of biasness, that is known as the ROB. So what it can do, it can go for the randomization, it can go for the missing out data, it will go for the selecting reporting. So it is just like an AI where it can tell you more about that, where you are missing, what is things that you have dropped out, what need are filled to have a desired outcome, whether you are missing or not. For example, you are feeling thirsty. Okay, you have a feeling. Now, what to drink? Cold drink. Okay, water, coconut water. This is the difference. It will take a decision to fill. He will choose water to make you feel okay. And when you want little energy, it will go for coconut water. Okay, then and when you want for a recreational, okay, drink, okay, for recreational mood, I can say, then you will go for this part. Now the next part I will tell you about the NOS. This is all about the selection, comparison, and the outcome. It will show you based on the stars. The grading criteria is based on star. So how many stars? Just you have a system known as in your gradation, in your examination, or you can say CGPA, or you sometimes say star mark. Okay, that whether he's got a star mark in examination or not. 80% marks or 85% marks, star mark. Okay, first division, second division, third division. So likewise. So if you're getting less than 5 stars, that means low quality. Greater than 7 star, that is high quality. 5 to 6 is the moderate. Okay. So these are the way you need to improve the quality. Now you need to go for the data extraction and the synthesis. You can go from the standardized data extraction way. So whether you can go for the study details, population, intervention, outcome, result, quality. So I've already, these things I've already discussed. So now the qualitative synthesis. Now here we will go for either we'll go for the narrative synthesis, whether we'll go for the thematic analysis, okay, or whether we are going for the swim, that is synthesis without meta-analysis. No need for any hardcore statistics. Getting my point? So you can have a 3 type of approach when this is known as the heterogeneous approach. Okay, where you have a multiple data, multiple aspect, multiple themes, multiple contrasting results, as well as your for your quantitative data. You can go for these things. So there are tools that are already given. That is the Excel sheets that you can use. You can use sheets from the Microsoft. You can go for NVivo for the thematic analysis. Revman I've already analyzed. I've already already provided you the name Revman for the meta-analysis, and the AP reviewer if you have a mixed approach. Okay, just I've given you the mixed approach, so you can go for this. Now this is one of the most important part, that is the artificial intelligence, okay, where we can go for the systematic literature review. So why we want AI intervention nowadays? Okay, because there are 4 aspects of AI. One is the big data explosion. There will be massive data, and it is very complex for a person to do a traditional analysis. Okay, you cannot go for just research, collection, networking, storage, then it's visualization and analysis. That is why for the big data explosions, you can use 3 different type of tools. That is the Talent, Apache, and the Rapid Miner. These are having data preparation, machine learning, distributed storage and processing capability, as well as data integration. Okay, so therefore these are the tools that will help you in the big data management. Now again, thing is the speed and efficacy. Your supervisor will tell you, no, we don't have 1 week. We have a deadline. You joined late. You have only 4 days to do. We need speed as well as efficacy. So AI can definitely help you in the screening of the article, formatting of the references, as well as analyzing the large databases. I was telling you. Okay, Zotero can be used for the reference management. Okay, RAN for the systematic review article screening. Elicit will be for your research assistant for your literature search. It will search all the type of literatures that are being available, as well as you can go for the size-based Copilot. Copilot means it will understand and it will explain the paper. For example, see, after the abstract, we went for opening of the full text. Now, how many full text you will read? Okay, it is impossible for you. 4 days it is impossible. When you will see, when you will do, when you will write, when you will submit. Okay, so go for the size-based Copilot. These tools are freely available, so go for them. Okay, it will provide you all the insights, highlights, everything. Just like people who have submitted, who used to submit paper in Elsevier. In Elsevier, there's a criteria that whenever there's a manuscript, there should be 5-point highlight. Okay, it will give you a 10-point highlight of that. 10 points it will give for each paper. That what is the gist of that paper? Okay, it's all about that. It's not about that abstract 250 word you need to read. It's just about some of the points it will provide. Now we have connected papers. This is the most favorite thing I have. For example, there is a chalcone paper. Okay, chalcone anti-inflammatory. Okay, now how many paper of chalcone anti-inflammatory are there? Now you can see there is a big dot. It is connected to one dot and another dot. So it will say that there are 4 paper which are very similar, and there are 15 papers which are closely related, and there are 20 papers which are having a chalcone term, and there are some papers which don't provide you a very good aspect of chalcone. Therefore, bigger the dot, bigger is the probability that it is matching to your area. So this will help in two ways. While writing a review, you should know that which paper are correlated to my subject. Therefore, you can reduce the repeatability of those aspects. Okay, that we are not repeating the same paper. Okay, and another approach is you can read those papers which are being associated with your paper. For example, chalcone anti-inflammatory. Okay, so this will give you both type of thing: where to go, where not to go. Okay. Now we have R Studio, that is for the data analysis and visualization. Okay, this is one of the best part where you can go for automate your statistical analysis. Now I told her that we people don't have that idea about biostatistics. It will provide you. If give the data, he will say 140 test. He will say ANOVA. He will say etc. etc. We need to do this. 2-arm, 1-arm comparison. It will be suggested by R Studio. So R is a free. Okay, so R Studio is paid, but R is free. Okay, you can go for the Python also if you want to go for machine-based learning. Okay, the statistical modeling, machine-based learning, and data cleaning. Okay, these all automated repetitive analysis steps can be done on a huge large database. Okay, Rightful is one of the best part. I will tell you that it is an AI powered writing assistant for scientific writing. So if you want that, you are given data, you don't have time to write systematic review, simply say, Rightful, please write based on the data I am giving you. It will write the whole paper for you. Okay, go and submit the paper for your publications. For the teachers, I can say, specifically for the teachers, I can also say that you have publication targets also. Okay, so these publication targets can be fulfilled, okay, using these type of AI tools. And to be very honest, it will not come under your AI flag detection of the Turnitin also. Okay, I don't know how it happens, but it happens. Okay, you will get either very low AI, or you will get zero AI. It will show asterisk mark, asterisk percentage AI report in the Turnitin. So if you are using a Rightful, okay, software. Now we'll go for the automation and accuracy. So NIME is a software for the data analytics. So just as you can see that it is connected, you can see from the logo that it is connecting. So it is connecting those papers which are diversely present in the database. So this is very important. Orange data mining. So this is just a drag and drop. Just drag your topic. Just drag your topic. It will do whatever the things about the data mining and learning. It will do. Go for the data robot. It is AutoML. Basically, it is automated machine learning. It will automate all the features, and it will give through the hyperparameter tuning method. It will give you a very important part, and that is what? That you will have a very close data. Earlier, what we have 1217. How many paper we are taking? 4. It will go give you 1 million paper, and you are taking only 2. 2 only paper of your interest from 1 million literature. Data robot will do it. JSP is a statistical analysis software which will do your t-test, student test, ANOVA test, your hypothesis testing, and everything. This is also a free way having AI. So it will provide you wherever the mistakes you are committing. Just like in ChemDraw, if you draw a wrong bond, it will give you a red mark. Okay, so it will just give you a introduction that, okay, you're doing it wrong here. Your data is not matching with the mean median mode table, just like that. So now we'll have an interdisciplinary demand. Nowadays, many of the studies are into different domains, and that is the reason you need to know about all those aspects, all those criterias, okay, about all those AIs. It will help in the bridging the knowledge and enhancing the collaboration across them. Okay, nowadays biomedical science. So people from bio, okay, mathematics and many others, say all are collaborating under one roof. So Slack, Trello, and I can say that this is another part. Okay, this is the Trello's advanced version, which is being used with latex document. Okay, we are using docs. Okay, many of the country researchers are using latex documents. Okay, that is very useful for the editing because latex can be is supported in Ubuntu also. Latex can be supported in Microsoft also. So if you're going for a if you're going for a team, okay, you can go for Slack so that you can communicate, because somewhere if you're going for a field study, if you want a field study based, okay, you want to do analysis, then also you can utilize Slack, where you can have 4 or 5 people. See, in that part, I can say in earlier where I was showing that pyramid, first part I was showing that we need to formulate a team, including biostatistician, including a person who will be doing a literature, include a person who will be going to write things. So Slack can be used from the first day. From the first day, you can use a Slack. Trello will be for the project management. Now, your supervisor, this is the supervising part. Okay. So he will assign the task. He will give the responsibility to the team member that what will be the task, how much time will be there, and where to stay organized with their timelines. So this is very, very important. So next is very small part that I'm going to go for, that is the bibliographic perspective. This is the last portion I'm going to explain you, that we need to have a good bibliographic referencing for the academic integrity, because we are not writing fiction story. Okay, we need evidence. If you are talking about that this specific disease is occurring due to eating this specific food, we need to have a reference. We cannot give a hypothesis. If you want a hypothesis, you need to do a systematic literature review. Got my point? Getting my point? That if you want to go for a hypothesis, then do systematic literature review and go for that systematic meta-analysis. Okay. And if you want that, at least I want that this is the particular thing, okay, this occurs due to this reason, we need to provide references, in-text references within bracket, and the complete references in the last portion of your paper. Okay, so it will help you in getting your credibility. So now you know it very well that whatever the things, author, publication, year, title, source, volume number, issue number, page number, and DOR and URLs. So these are the things which are pretty important. Now you know about various styles. We use more Vancouver as well as APA. Okay. Sometimes we generally use MLA also, but too restricted. But Vancouver is the most popular for the science. Okay. So therefore, I'm not going to explain you that how we need to write and things. There are some tools. I can say there are 3 parts. One is the reference manager, that is the Zotero, EndNote, and the Mendeley. These all are the free. However, EndNote is a paid version. Okay, but Zotero you can use for free. Mendeley you can also use for free. There is no issue. Then we'll go for a citation generator. Okay, this is what citation generator. For example, if you want a particular study, it will search the particular study and there will be a drop box. There will be a drop box just like that. There it will show style. So once if you click the drop box, it will show APA, MLA, Chicago, IEEE, Vancouver. Once if I choose Vancouver, it will show me that reference in Vancouver style. Okay. So MyBib as well as Google Scholar. In Google Scholar, there is a quotation mark. Okay, click that quotation mark. You will be getting the same thing. Okay, in many type of ways you can go for the same literature. Okay, so MyBib is a free, and actually we need a BibText to support it. Okay, support system. So last one is a word processor plugins. So you can use that MS Word plugins for to add, just like you have now you have added Grammarly. So therefore, if you are using LinkedIn, if you are using uh, Facebook, if you are using any other, now it will go there and it will correct the things. Okay, getting my point? So that plugins are very important. So now you can have the references plugin so that it can import from the source. Okay. So this was the site is for me. This is the website. You can go for the create citation, and you can also check for the plagiarism. So this is one of the area where you can trust for the citations. This is a Zotero. You can see that you can have the references. You can provide that each and every. Okay, you can add. Okay, all kind of papers, all kind of newspaper articles, or type of snapshots, links, whatever you want. You can go for. If it is a unfilled item, it will go to a different folder. If it is a duplicate item, it will automatically go. And if you can delete from it, it will go to trash, and from trash you can delete it. Okay, if you don't want. So this was your Mendeley, where you can get a lot of citations from the author information, from year to title source, okay, to everything. When you have added and what is the file. So you can have a reference manager best as a Mendeley. So before finishing it up, I would just like to conclude that the systematic literature review, it is the most rigorous and unbiased approach. So the pharmacy people, particularly pharmacy people, they must use this structured methodology and protocol driven approach everywhere. So now you have some tools such as RAN, Zotero, as well as Revman that you can use for the literature review. You can have a database such as the Cochrane you can use. Okay, if you can use the quality assessment parameters that I've already told you about the Ottawa protocol as well as Cochrane risk of bias, okay, ROB. Then you can use the emerging technologies like AI assisted screening that I've already given you, AS review and the elicit, okay. And you can go for a collaborative approach because you need to have a team, okay. You need to have a team, that is most important, okay. First, you need to have a team, okay. The first, then you can formulate a query, okay. That team can be managed, that team can collaborate, that can continuously update, that can do many things, okay. This is the best way. And I can also tell you that there will be some research questions, there will be some protocols, and that will be effectively being managed, okay, through this systematic review AI assisted domains. And if I talk about academic value, you can go for the policy decision, you can go for the clinical guidelines, you can go for all kind of I can say utility in the society. Nowadays, you are getting some projects. Projects are call for projects from government. Now, government is asking for that how you can apply in the social level. Now you have approach like I will do a synthesis. Now, majority of the screening person who are sitting in the government, they are not finding any immediate translational ability of your project to the society. Okay. So this systematic review can provide you an approach, an area where you need to go for. If you have a previous publication, see nowadays you need to provide that there should be some evidences. Okay, just like ANRF was there, na? ANRF in ANRF also we need to go for the previous work done so far as well as the previous concepts and whatever the publications you have done in that area. Okay, so this is one of the best way to get a funding as well as approach towards the project from a government agency, that you can go for systematic review. People will trust, people will cite, people will adore your work, okay. And those works, because see, people when they are going for screening of the project protocols, okay, they will simply, okay, they will simply be going for, okay, whatever the far, okay, to the society, to the scientific community, that will give additional weightage. So systematic review can give you more weightage, okay, if you are planning for certain projects, because immediate effect can only be provided through systematic literature review, and publishing those reviews can give you as much as weightage as a research, okay. So I can just say that I will be very thankful to the Honorable Chairman, sir, of the ICare and the secretary of the ICare, all management professionals, to the principal of Haldia Institute of Pharmacy, and the coordinator of the FDP, to all person of the teaching staffs, to all non-teaching person who is working hard to make this event successful, to all delegates for their patient listing, all participants for their, I can say, uh, I hope you have enjoyed a lot from the session, and all the young minds. Okay, so those who have already been there as a volunteer, as a participant, as those who have come with their interest. So I convey my heartfelt thanks to all of you. So, so if you want any future collaboration with me, I am always ready. So you can find me on all social medias or on scientific platforms. These are my identifiers. You can see from the Web of Science to Scopus to Publons to everywhere. Okay, this is my profile at present. So you can simply drop an email at my official emails or even at the personal emails. Okay, if you wish, and my contact number is also there. So I'm also serving currently as the director for the Suktas Organic and Life Science Private Limited. That is a startup company. So anywhere, anything of your interest, if you want to collaborate, I would be most happy to be associated with you all. So I can say a very thanks to everyone for the patient listening, and I hope you enjoyed the session a lot. Thank you.
Thank you, sir. Your session has added immense value for this program. And thank you to all the participants for having patience. And I would like, if anybody having any query, questions, you can ask to speaker, sir. Just unmute yourself and you can ask the
Yeah,
question.
sure. Yeah, sure. Please ask. So any question, any query, any suggestions if you have. I am also looking forward to suggestions.
If you want, you can drop your questions in the chat box to us.
Suggestions, okay. Yeah, in the chat box, if you want to give some suggestions, if you want to tell something, please.
Okay, sir, since we have a feedback form, so if we got any kind of suggestions, we'll definitely share with you. And I'll request Panda sir to be in contact with you for any
Yeah,
feedback
definitely.
suggestions if we
And
receive from any
please
participants.
do share the picture of the session. I would love to post and I would love to share my experience on social media about this session. And I would love to associate with Haldia Institute of Pharmacy in future also.
Sure, sir. Thanks a lot, sir. Thank you so much.
Thank you, thank you.
Now
Thanks a
I
lot.
would like
Yeah.
to call Dr. Lagnit Mahapatra, sir, to please have a vote of thanks to Devashishan. Please, sir.
So good afternoon, everyone. I'm Dr. Lagnit Mahapatra. On behalf of Haldia Institute of Pharmacy, I like to thank to Dr. D.K. Mahapatra sir from Chitkara University for his valuable speech and sharing his knowledge about the literature review, which definitely help us for the research and definitely help for the participants, all the participants for their research works. And with this, uh, on our principal and management members, definitely I'm thankful for them for their continuous encouragement, and also I'm thankful for the coordinator of this program of this session for his commitment and commitments what they have done making the event success. And finally, I'm thankful to all the participants for their active participation, for which we are making this event and this session successful. Thank you, everyone, and definitely it will be helpful for all. Thank you very much. Thank you, sir.
Thank you. Thanks a lot.
I'll request every participants. I'll just share the feedback form in the group. Kindly please fill it out. Thank you. And thank you, Doshi sir. Have a great day.
Thank you. Thanks a
Thanks,
lot.
sir.
Thanks a lot.
Sir, you can leave, sir.
Yeah. Thank you. Thank you,
Thank you,
everyone.
sir.
Thank you
Thank you,
for
sir.
patient listening.
Pardon, sir?
No, I just thank everybody for their patient listening. So I am leaving. So it's a nice experience. So thanks a lot.
Thank you, sir. I hope we'll see you again, sir.
Yeah, sure.
Thank you, sir.
Thank you.